I'm going to talk to you a little bit about a long-standing and long-running project in our lab, and that project is called DipFish, and it really arises from our long-standing interest in being able to spatially map the positions of RNA, and the tools that have been developed around microscopy, spatial mapping, and computational tools have made it possible even for us to contemplate the ability to actually use image-based approaches to be able to count the numbers of RNA molecules and protein molecules at single cell level. Just so I say these things in case I run out of time, this particular project has been supported by the Chan Zuckerberg Initiative, and we have been a lab that has received support from the CZI to perform this work. So moving on. So spatially resolved omics really was very well encapsulated in this review from Alexander van Nieuwenhoorn's lab. And it's really the ability to be able to map at single cell resolution and subcellular resolution the positions of RNA and protein. And doing so has been a long-standing desire of scientists, because most of the work that has been done to be able to count RNA molecules, especially, and protein molecules, has been done through biochemical approaches, which are highly destructive. So essentially, what we've ended up with is techniques where, to actually count the number of RNA molecules, for example, the cell itself is destroyed, and a big number of RNA molecules that survive that destruction process are counted, and they're assumed to be accurate. However, we know that biology takes place in 3 dimensions, and so the absence of the spatial information of where these RNAs and proteins, these central dogma molecules, are located, has a very significant cost to our deeper understanding of the roles that these molecules may play. So yes, it's important for us to be able to count how many molecules of RNA they are and how many molecules of particular proteins they are, and that'll give us information about what cell state and cell type that is. But actually, as I'm going to show you through this talk today, the cellular state is probably more informatively seen through the spatial distribution of specific RNAs. So recently, some members of our lab have published a really nice commentary in Frontiers in Cell Biology, and this is a figure reproduced from there, where this process of identifying cellular contents, RNA here in this example, for single cell sequencing is contrasted with what could happen when you do the same experiment by spatial transcriptomics. You can see here that we have the dissociation process I described, RNA isolation and reverse transcription and amplification. I think from Aviv's talk, and many of you already have a lot of background on how the process of single cell RNA sequencing occurs. And that in turn gives us either TSNE or UMAP plots, and this dimensional reduction allows us to see different clusters of cells, and these clusters of cells are essentially telling us over here that this particular cluster corresponds to a particular cell type. Now, the challenge is that within this particular cluster, we may have cells that exist in actually these 3 different states, where the RNA of a specific transcript, and even though these transcript numbers are essentially the same, will be localized in multiple different positions. And that localization in multiple different positions may correspond to different cellular structures, different cellular markers, or even distinct different cellular states. And this, unfortunately, isn't captured by single-cell RNA sequencing, and therefore shows us most poignantly how understanding the spatial distribution of RNA is highly important to our understanding of single-cell biology. So many techniques have emerged to be able to sort of decorate RNA, and these methods range quite extensively from from hybridization probes like single molecule FISH probes that came from the lab of Sanjay Tyagi and Fred Kramer. And these are essentially small oligonucleotide probes labeled with fluorescent dyes that are tiled along an RNA. And the example here, which was first performed by Arjun Raj when he was with Sanjay Tyagi, was really a beautiful example of how using hybridization probes, we can discretely decorate individual RNAs with specific fluorescent signals. Now, these particular probes can be sort of remixed, if you like, into different types of barcoding strategies. And perhaps the most famous of these strategies is the Murphy strategy, which uses a Hamming code. And this Hamming code allows the use of multiple color probes, which hybridize to different RNA species. But then by using a very carefully worked out registration where staining, imaging, and erasing of the fluorescent mark are done in a sequential process, you're able to measure up to hundreds of different individual RNA transcripts, in other words, different genes, using just a spectral barcoding regime of as little as 4 different colors. And this is really allowing us to contemplate the single cell RNA interrogation of multiple RNA species at single cell resolution. So other methods have been emerged as well, and I really won't go into these because these are kind of, let's call it minor methods that are used in the field, but one of them is TomoSeq, where you basically use fixed probes that you use for RNA sequencing in combination with tomography. Another one is called TivaSeq, where you use a specific type of tag that is then photo activated to be able to see different thread pairs, and this allows you to do multiple labeling and achieve spatial resolve transcriptomics. But more fundamentally, from a biological standpoint, why the interest in doing this sort of transcriptomics that allows us to reveal spatial localizations of RNA? Now, we know in eukaryotic cells that RNA is made in nucleus, and messenger RNA is then transported to the cytoplasm, and there it gets translated into protein. One of the things that has emerged, especially from developmental biology, is that this spatial separation of transcription and translation of messenger RNA doesn't just lead to sort of RNA exiting the nucleus and being present anywhere in the nucleus to be translated. But in fact, developmental biology has revealed to us really interesting regimes where RNAs are trafficked for very long distances to specific subcellular locations where their subcellular location is highly influential to developmental processes. And here, using a technique called molecular beacons, the red RNA, which is the red molecules, which are RNA, are decorated with these molecular beacon probes, and they're able to show this long-range transit from this nucleus here in one of the nurse cells of this oocyte all the way to the posterior region of this oocyte, where this particular RNA called Oscar is going to be able to determine the posterior part of the future embryo. It's also going to be highly influential in the formation of gonads in this future embryo, fly embryo, Drosophila fly embryo. The green molecules are the protein staufen, which is thought to be involved in the very late stages of RNA localization of Oster. And so you can see here that this is a poignant example of how localization of RNA to specific subcellular locales is highly important for developmental processes and is assisted by the interactions of different proteins. So being able to understand this has led us to see that actually this spatial separation of transcription and translation is not only important in, say, the developmental stages of different multicellular organisms, but it also turns out to be a basic feature of general homeostasis in multiple processes. So here, I'm just showing you different examples in, say, neurons, where the localization of specific RNAs to dendrites is highly important to the formation and plasticity of neurons and formation of long-term memory. Or here in this beautiful paper from Moore et al. In Science from a few years ago, shows how depending on the state of these villi cells, there's an accumulation of specific species of RNA at the apical site or the basal site, depending on when this animal, in this case it's a mouse, is actually feeding. So the accumulation of ApoB messenger RNA at the apical part of the enterocyte is part of the process of this, the feeding process, the actual activity of feeding of the mouse is highly influenced by this spatial position of ApoB messenger RNA. And of course, there's some seminal examples of unicellular organisms visualized with one of the earliest techniques to be able to visualize RNA in live cells, which is MS2 RNA. And this is from Roch Singer's lab. And this experiment was done by Edouard Bertrand, published in Molecular Cell, where ASH1 messenger RNA is shown to accumulate at the bud tip of a yeast daughter cell and is being imaged live in this technique, again showing the importance of the subcellular localization of messenger RNA. This is another image here from a study in rat hippocampal neurons, which shows the accumulation of messenger RNA after immediate early gene expression in different nuclear bodies and at different dendrite tips of these hippocampi. Again, a key process involved in memory formation, highly determined by the spatial localization of messenger RNA. So one of the first things that we sought to do was to develop assays that could quantitatively determine RNA localization. And one of the easy ways of doing this is that in developmental organisms like the examples I've just shown you, you have highly polarized cells, and this makes it quite easy to be able to determine RNA localization because of the inherent polarization of these structures. However, ordinarily, most of the cells that we work with aren't really polarized. And so we have to sort of reconstitute these cellular microenvironments. And one of the ways to do this is by a technique called micropatterning. And micropatterning has this distinct advantage in that it gives us the geometries and architectures of intercellular interactions by being able to fix cells into a specific geometry in cell culture consistently. Now, what do I mean by that? Well, if you use micropatterns, which essentially pattern the surface on which we grow cells, this allows them to reproducibly form specific shapes. And this kind of benefit of this is that this reproduction of specific shapes by cells allows a highly reproducible internal organization to occur. And that's really important because it shows us that even though cells appear to grow willy-nilly, say in culture, the actual internal organization is highly controlled and dictated by cellular processes and is very highly reproducible. And one of the most important determinants of that reproducibility is the shape in which the cell will be grown. So this work by Manuel Thierry and Michel Borsans has shown how being able to grow cells on these microfabricated micropatterns forces them to grow into specific shapes. And by forcing them to grow into these highly reproducible shapes, if one looks at the internal organization of these cells, one sees that consistently specific cellular organelles and cellular structures always reproducibly accumulate in specific regions, subcellular regions of the cell. And here from this study, also from the Curie Institute, we can see how specific cellular proteins involved in secretory pathways like RAP5, SEC13, and RAP6 reproducibly accumulate in distinct regions of this micropattern cells. And this is suggesting to us that proteins, subcellular organelles, all accumulate systematically in specific regions. And this begs a much deeper question, which is, does RNA do the same thing? In other words, if changing the geometry of cells makes all their internal cellular organelles reproducibly localized to the same areas, does that same event cause RNA to reproduce its local spatial distribution consistently and deterministically from cellular shape? So we sought to sort of answer this question, and we used these probes that I mentioned before, these single molecule FISH probes, to be able to label different RNAs and do so on micropatterns and look at whether their organization and their spatial distribution would localize in a highly reproducible manner. Now, obviously, you could argue that it would be as simple as us staining with single molecule FISH for these RNAs, but actually, the process of doing this was a lot more complicated, not necessarily because of the way we performed the experiment, but because of the way we analyzed this data. So we took standard cells and using fibronectin micropatterns, we put N3T3 cells onto these micropatterns and then stained them with single molecule FISH probes for specific RNAs that we knew in the main would adopt specific localizations upon stimulation. And what we had to do this experiment is we needed to devise some new computational and image analysis tools that allow us to initially detect these spots, align multiple cells, since they all have the same shape, to a shared nucleus centroid, and then use different 2D and 3D segmentation processes to be able to segment the image into different channels. And after that, we detected subcellular structures and organelles within these cells to be able to know how the distribution of these RNAs related to these cellular organelles. So in other words, we would use structures like the MTOC as cellular anchors to be able to determine where the RNA was relative to these cellular anchors. And so one of the key parts of this was that in our initial studies, being able to do single molecule FISH necessitated using high power objectives. So we had to use objectives that were these 60x to be able to detect these single molecule RNA FISH spots. However, that would pose a problem in itself because we would have to assay hundreds of these cells, and we'd have to do this in a highly automated way to be able to acquire the necessary data amounts to be able to draw important and relevant statistics. So we developed some computational tools that could control microscopes, and this controls almost any microscope in the market, which can automatically detect micropatterns and automatically detect patterns which are occupied by cells, and then take full control of the microscope and acquire images in an automated fashion and analyze these images and perform the segmentation tasks that I've just mentioned to you before. And once we performed this, we were able to do some standard analysis. And one of the things we saw that about micropattern cells versus normal cells is that in micropattern cells, the deviation in the distribution of RNAs in the cells was reduced after a very low number of cells. So this is a measure of absolute deviation, and at about 10 cells, the deviation drops dramatically to about 80% consistency across all the cells. To 0.8 consistencies. This is an arbitrary measure across all the cells. And when growing cells in culture, this type of absolute deviation can never be reached. In other words, these low levels of deviation cannot be reached. Even if you continue to acquire more and more cells, the sort of random nature of their organization means that there's high deviations in spatial organizations between cells. We also could measure other things like mean squared error of the number of cytoplasmic cells versus cytoplasmic spots versus peripheral spots, and the spread to which RNAs make. And again, you can see that micropattern cells possess distinct advantages in their ability to reproduce these all these parameters across different subcellular measurements in cells. Another measurement that we did undertake is transcript number. And this is important because it leads back to the idea of doing spatially resolved transcriptomics. What we see here is that we have very high variation in the numbers of transcripts for a given cellular size. So you can see here that the variation in transcript number on the left here for standard cells in culture is extremely high, especially when compared to micropattern cells, where variation is far more reduced at a given size for a micropattern cell. So given two micropattern cells that are patterned on the same size patterns, the deviation between these cells, the number of transcripts that's being made is extremely low. So we used this analysis to start to study the localization of different RNAs. Some of these RNAs we knew would be localized to the peripheral fraction of cells upon stimulation with, say, fibronectin, while others we did not know of their localization, and those were used as kind of interrogatory controls. And even further, others that were used simply as controls that would be used in qPCR, which we knew in general had very random distributions. And so you can see here, we're measuring the distance from the nucleus all the way to the periphery. And you can see specific RNAs are highly enriched at the periphery, while others have far lower enrichment at the periphery. And this enrichment at the periphery, we were able to delineate into the 30% fraction and the 10% fraction. And some of these RNAs have a very high localization just at the 10% fraction. In other words, this very thin outer edge of the cell, as they seem to play a very important role in the polarization process. And one of those was PAR-D3, which seemed to have a very not much localization to the periphery until the last 30% and even 10% fraction, where there's a dramatic increase in its localization to the periphery of the RNA. Now, to be able to do clustering, we built upon some prior work that had used different types of clustering approaches, and we basically developed two models of clustering. The first one was a complete spatial randomness model. In other words, could we fit RNA distribution? Because perhaps not all RNAs localize in a deterministic manner, and their localization is purely random. And so these would have to fit a complete spatial randomness function. And both of these we tried to model around a Ripley square algorithm. The second type of, let's call it clustering or distribution that we would expect is spatially clustered. So you can either be completely spatially randomly clustered, or you can be spatially clustered. And the assumption is that if you're spatially clustered, that clustering is deterministic. And so there's two different functions that we can fit any RNA distribution. You either are completely spatially random, or you are spatially clustered. And that spatial clustering with the by Ripley K score could tell us precisely the degree of clustering or spatial clustering that's occurring. And this could be done for an RNA, and then it could be further translated into a function for a protein, spatial clustering, or a protein that's completely spatially random. So we would follow after stimulation, the position of different RNAs. And these RNAs, the same RNAs that showed you when we looked at the distance, and they would travel from the nucleus to the periphery. And here we're looking purely at the degree of clustering. And in the solid line, we're looking at the RNA clustering. And in the dotted line, we're looking at the protein clustering. Now, one of the most important things about this is that if you look at the RNA clustering, you see that the RNA clustering first clusters prior to the protein clustering, again, consistent with how we understand that RNA transcription precedes protein translation. And so, sorry about that. So what you can see here is that you have, for example, Ardea, which clusters in the early time points after stimulation by fibronectin. And then at the later time point, you have protein clustering. Likewise, you also have GAPDH, which is one of our controls, which never clusters. And that never clustering means that we get first the probably random distribution and completely spatially random distribution of RNA from GAPDH, which may pass, say, through the ribosome and then be made into a protein, but that protein also gets completely spatially distributed. I mentioned party 3 earlier, and we know that party 3 RNA takes a great deal of time before it reaches the periphery, but there it clusters very heavily, and likewise, party 3 protein also clusters 3 or 4 hours later after the RNA clusters, presumably in the same location where the RNA is clustered. So you can take this data and now relate it to how cellular organelles are positioned during this polarization process. And the way we did that is we divided cells into different quadrants with MTOC labeled, and then we were able to relate the position of the MTOC to where specific RNAs were located. Now, the reason we use the MTOC is because the MTOC seems to be very important in determining the center of the cell. And what we came to understand is that there are RNAs that reposition with the MTOC. There are other RNAs which reposition away from the MTOC, and positioning away from the MTOC or with the MTOC seems to imply that the MTOC, or the center of the cell, has a very important and deterministic role to play in how cells sense their positions, like how RNAs sense their positions in terms of the cellular landscape, there's this cellular distribution. So this is highly important because the fundamental question we have is that if we're having reproducible subcellular localization of cellular organelles and of cellular proteins, as is being described in the literature, there has to be some object that these central dogma molecules sense to be able to determine their localization vis-a-vis a center of gravity of the cell. And so for us, the MTOC was the strongest candidate. And using this information, we derived something called the MPI, which is the MTOC polarity index. And we could see that, for example, Ardea RNA had a very high MPI, which means that the RNA is highly sensitive to the position of the MTOC, whereas the protein is not. So this is telling us that the RNA around 5 hours becomes highly sensitive to the MTOC position, and that in turn seems to determine where the RNA will be subcellularly located. And that's then followed by protein being made where that RNA clusters. I showed you the previous slide. And that clustering seems less dependent on the MTOC position. We see similar dynamics with PAR-D3. Now, does this mRNA position influence protein position over time? That's a really important question because we know that RNA position, for example, in neurons, determines where proteins are made, say, near the postsynaptic density in a stimulatory manner. And so an important quantitative measurement that we need to be able to do at single cell level is to be able to know where an RNA is in relation to where a protein is. And so we developed this tool called a temporal index. And this temporal index, or TIS, temporal index score, this TIS, was derived in the following manner. We would take RNA positions measured by single molecule FISH, and we would take protein positions measured by immunofluorescence, and we would then divide the cell into multiple subareas and then assess the messenger RNA and protein per subarea sequentially. So you can see here, we've broken up the entire cell into different positions. And here in each subcellular location, we are relating the position of an RNA and a protein to each other by their Euclidean distance. And then this relationship can then be measured by the tools that I've previously shown you before. In other words, whether they are in complete spatial randomness or whether they're spatially clustered, and then looking at how closely the RNA and the protein are in distance to each other. And then these measurements taken over time can then relate this temporal interaction score for, say, beta-actin may be quite high because that's showing us that there's a high relationship between the RNA and the protein over time, or say PAR-D3, where that relationship can be quite low. So when we look at two examples of how this TIS measurement can occur, we look at GAP-DH, we see that the temporal interaction score is high at early time points and then becomes lower at later time points. And this cartoon here is sort of showing us how that looks in terms of imaging, where we know that the RNA of GAP-DH is made, it goes to the ribosome, it then makes GAP-DH protein, and then it seems like that RNA just dissipates in a basically highly random approach with a lot of unclustered RNA accumulating and distributing throughout the cellular volume. And that's seen a bit larger here of that process in terms of these three stages of this process for GAP-DH. Whereas we can look at beta-actin, and here I can just point to the cartoon where beta-actin RNA is made. And we know beta-actin RNA is really central to the polarization process and seems to accumulate in the periphery. And that accumulation becomes peaked at about 3 hours, where we start to make beta-actin protein, and that beta-actin protein corresponds to clusters at the periphery, and those clusters at the periphery also correspond to the clusters that accumulate of beta-actin protein at the periphery and are essential to the mobility and the polarization of these cells. So we can see these two distinct measurements can be taken in this cell type. Now, if our quantitative approaches to spatial transcriptomics are accurate, they also mean that perturbations to the cellular architecture should be measurable by these techniques that we've derived. And indeed, that turns out to be the case. One of these perturbations is the addition of cortisol, which perturbs the MTOC and also perturbs the microtubule organization of cells. And we can see here that when we measure the MPI the MTOC polarity index of ARGDIA RNA. Remember, I showed you that this MTOC score, MPI score for ARGDIA was very significant. And indeed, when we add cortisol, we can see a significant change in that MPI score, suggesting that the disruption of the MTOC in the microtubule architecture is highly influential to how ARGDIA messenger RNA is able to sense its position within cells. And when we look at that by imaging, what we can see here is that ordinarily ARGDIA congregates with MTOC as it can sense the position of the MTOC. And in the presence of cortisol, that ability of the ARGDIA messenger RNA to co-localize the MTOC is lost. Interestingly and contrastingly, PAR-D3 in the presence of cortisol, which gains a much higher MPI score, where prior to the addition of the cortisol, it did not have this score. And this is indicated in images that we would acquire here in this cartoon, where PAR-D3, as I mentioned earlier, accumulates in the periphery at a very late stage in its migration to the periphery, and there the protein and the RNA accumulate. In the presence of cortisol, it seems that that accumulation becomes even more highly dependent on the MTOC, the RNA. And this all points to very interesting biology, because you can imagine that these techniques can be used to interrogate the cellular machinery that's most influential to the transport and localization of these messenger RNAs and proteins. And that could be the actin network, or that could be the microtubule network. So how could we use this, say, in our ability to build atlases with spatial transcriptomics? Now, we applied this to a long-standing question in muscle biology, and that long-standing question is how muscle cells, which are very large, are able to form these reproducible Z lines in multi-nucleated cells. So this cell has multi-nucleated, and it seems to form these periodic Z lines. And these periodic Z lines are essentially parts of the sarcomere that are combined together to form the skeletal muscle fiber. This is a really interesting problem, and we collaborated with the lab of Edgar Gomez at the IMM in Lisbon, and they are world experts in muscle development, and are also have one of the few labs where you can actually develop these skeletal muscle fibers from mice ex vivo. And so one of these long-standing questions around the formation of these Z lines is how these multi-nucleated cells are able to position the Z lines at specific locations. And so the question is the influence of each of these nuclei in forming the domain and making sure that the Z line is correctly positioned is actually a really, really intriguing question, because there are no membranes that separate these nuclei, right? So how can it determine the position of the Z line? Since the Z line falls in between, these Z lines fall in between these nuclei. So one of the theories is that maybe the Z line is determined by the RNA localization of specific proteins that constitute the Z line itself. And so we looked developmentally at immature muscle fibers to mature muscle fibers in vivo. And in these immature muscle fibers, we were able to stain for actin 2 messenger RNA, and at the same time stain for F-actin protein. And in blue here, you can see where these nuclei are. So there's a nucleus here, there's a nucleus here, and it seems that a periodic number of F-actin lines, protein lines, form between these two nuclei in the immature muscle fiber. So if we look at this particular area, you can see in the immature stage, we're seeing these sort of F-actin proteins accumulating. And as the muscle matures, these F-actin proteins are becoming more dense. And in fact, with time, they start to form very solid lines, which are important to the integrity of the muscle fiber. And they form these lines between the two nuclei of this very large muscle cell. At the same time, you can see here that the actin 2 messenger RNA in the immature muscle fiber seems to populate areas that encompass not only the green areas of F-actin, but the intermediate areas. And as the muscle matures, these actin 2 messenger RNAs seem to also co-localize with the F-actin and exit the intermediate areas over time. And that's contrasted here with GAPDH messenger RNA, which seems to have no general accumulating spot. And we can see this example here where we computationally impose a mask of all the Z lines, all the F-actin lines, and we impose a messenger RNA spot detection mask to see whether these two will overlap in time. And so what we find is that when we apply the dip-fish algorithm, we can see that there are no periodic intervals, even after 80 divisions, and there is no clustering of the messenger RNA. And the temporal interaction RNA and the protein is actually lost or not present. And when it is not present, there are no Z lines formed. However, as the muscle matures, you get these bands, actin 2, that begin to form between the nuclei. And these bands of actin 2 begin to form at distinct periodic distances. So the actin 2 messenger RNA seems to accumulate in periodic distances between the two nuclei. And as it does so, the F-actin seems to be accumulating exactly at the same period of this actin 2. So this is again suggesting that actin 2, by accumulating in this periodic manner, makes a protein on which the F-actin can accumulate to form these Z lines. And this is the first time that this hypothesis has sort of been tested to show that the RNA, the actin 2 messenger RNA, seems to be deterministic of where the F-actin protein accumulation will occur. That relationship is seen here to not be the case for GAPDH, where a similar periodic line doesn't form. So this is sort of a summary of that data here. So in the last minute or two, I just want to show you some new imaging approaches that we're using to be able to do this, use dip-fish tools in tissues. And those tools rely on a new type of optics called synthetic aperture optics. And synthetic aperture optics sort of violates Snell's law here by essentially making us have a very large field of view at a very high magnification, which ordinarily is not possible, and a very high depth of field. And that means, like, for example, in this tissue here, we can have an image of the tissue with a 20x objective, but then offline, we're able to zoom into individual areas here to be able to see each individual nucleus. And if we've stained not only the nucleus, but the RNA, we can also see the individual RNAs as well. And so here's an example of us using this type of imaging where we can have multiple viewpoints offline that allow us to look at hundreds of cells and then visualize individual cells for the presence of messenger RNAs. And we can count these at a single cell level, do spatial transcriptomics. So with that, I'd like to end. This is a picture of where our lab used to be in Cape Town. And I just want to thank our collaborators. And also this work that I presented today is the work of Anka Savulescu, a senior scientist in the lab. And she worked with Robin Bracken and Nicola Bohm, bioinformatician in the lab, to develop a lot of important aspects of the dip-fish algorithm as well as 2 data scientists in our lab, Andre Cures and Gabriel Tusu. So with that, I want to thank you very much for your attention.